This is podcast number 16. Okay, so we're still on carbohydrates. We did glycolysis and then gluconeogenesis. And now we're going to take that pyruvate that came out of glycolysis and finish oxidizing it and get lots more ATP than just 2 that we got out of glycolysis. So this is a diagram of a mitochondrion. Not a realistic diagram because, as we'll talk about in a couple slides, there's this huge anastomosing network, not a little kidney bean. And again, I don't want you to draw this sort of cutout diagram on a test, just a simple outline that shows me the two membranes. So here's the outer membrane here, and then inner membrane here. And pyruvate has to get from the cytoplasm across both membranes into the matrix. So across the outer membrane and the inner membrane and the intermembrane space, intermembrane space. There is no inner membrane space, but there is an intermembrane space with a T. Okay, it's across that space into the matrix, which is inside both membranes. And in the matrix, pyruvate is decarboxylated. CO2 is removed. This is some of the CO2 that you breathe out. It happens from this procedure. Then that generates an acetyl-CoA molecule, which gets added to a 4-carbon molecule in the citric acid cycle. It's decarboxylated twice, so that 6-carbon molecule becomes a 4-carbon molecule and then goes around that cycle again. That cycle makes a little bit of ATP if you're a plant, GTP if you're an animal. Mostly what it makes is these reduced molecules, NADH and FADH2. And then finally, you're going to take the NADH from glycolysis, which has to be imported into the mitochondrion, and the NADH and FADH2 from the citric acid cycle. You're going to oxidize those in the electron transport chain, stage 4. That's where you're going to use these high energy electrons get taken off. These electron carriers are going to be used to move protons into the intermembrane space, and that protons are going to drive a gradient where protons move through a channel that will use the gradient to spin a enzyme that will add ADP and P together to make ATP. And this is where you're going to get most of your ATP. Later on, we're going to talk about how you can take apart a fat molecule, and then those pieces of fat molecule can enter this part of the cycle. So we're going to see this citric acid cycle multiple times. Okay, as I mentioned before, mitochondria are not the little kidney beans, but they're a large anastomosing network of membrane constantly changing. And the reason we have this picture in our head of these is mostly because that's what you see in books. And when we take an electron micrograph, you're just taking a slice in time, a literal slice, as you slice through the tissue whenever that is fixed. But if you could watch them over time, you'd see that they change shape and position constantly. There are these experiments done with transmission electron microscopy where the specimen is tilted and then you take lots of different pictures of it. It's called a goniometer stage series, and you get different angles of view on an image. And by doing this, you get this pseudo 3D shape where this yellow is the inner membrane and the greenish blue on the outside is actually inner membrane, and then outer membrane right on top of that. Here is a more realistic picture of a mitochondrion. We see it more like a tube of stuff, and then these multicolored invaginations here are the cristae of the inner membrane. In this diagram here, there is no outer membrane shown on the diagram. This is just inner membrane, and the inner membrane at certain spots pinches in and fold these big folds. So this green is connected to this sort of fatigue green on the outside as one continuous membrane, and this light blue is connected right there to the inner membrane. All of this is inner membrane, and again, the outer membrane is not shown on this image. So compartmentalization is very important to cell function in general, eukaryotic cell function, and within organelles it's also important. There are different compartments of mitochondria. In the outer membrane you get phospholipid synthesis, fatty acid desaturation, and that's removing putting in double bonds. Fatty acid elongation, going from say 18 carbons long to say 26 carbons long. On the inner membrane, that's where the electron transport chain is. The mitochondrial inner membrane, remember, is one of the highest percentage of protein in a biological membrane, up to 90% protein by weight. And in the matrix, that's where pyruvate oxidation happens. So the pyruvate is going to cross two membranes, the outer membrane and the inner membrane, and then get oxidized in the matrix. That's also where the citric acid cycle happens. That's where you get ATP synthesized, and then ATP has to get back out into the cell. We're going to see beta oxidation happen in the matrix. The mitochondrial DNA gets replicated in the matrix, and mitochondrial RNA gets synthesized in the matrix, and mitochondrial protein gets made in the matrix. Okay. These little enzyme complexes, you've seen them in multiple classes. They're called FO, F1, not F0, but actually FO, because the O stands for oligomyosin sensitive. Oligomyosin is an antibiotic, and it works by poisoning this channel for protons in the FO, F1 pump. The F1 part is the enzyme, the synthase. So protons will move through this FO channel and bind to a complex on the F1 complex, causing this to rotate and causing ATP to be synthesized, and then ATP is released in the matrix. The F1 part of this complex, the FO F1 complex, is pointing toward the matrix. In mitochondria, it's pointing toward the matrix. In chloroplasts, it has a different orientation that is related to chloroplasts being swallowed up twice in endosymbiosis, two membranes, but we'll cross that road when we come to it. Okay, so outer membrane, porins that allow things to get in and out of the outer membrane into the intermembrane space. Intermembrane space, inner membrane with invaginations called cristae, and lots and lots of FO, F1 complexes on that inner membrane with the F1 part pointing into the matrix being here. Okay, so pyruvate came from glycolysis. It crossed the inner membrane and the outer membrane to get to the matrix. So pyruvate from the cytoplasm crosses both inner membrane and outer membrane, is acted on by pyruvate dehydrogenase, which is a matrix enzyme. So pyruvate has to get to the matrix, okay? And it's a giant enzyme. So it's called the pyruvate dehydrogenase complex. You can actually see it in an electron microscope. and it's a very large enzyme complex. Okay. Pyruvate gets decarboxylated, so 3-carbon molecule, one of the carbons comes off as CO2. You breathe that out. The remaining 2 carbons are acetyl group. They're attached to coenzyme A. Coenzyme A are not enzymes, but they are compounds that help enzymes work. Many coenzymes are vitamins. Coenzyme A plus acetyl group gets added to a 4-carbon group here, 1, 2, 3, 4 oxaloacetate, to make a 6-carbon citrate. Usually this is, in most diagrams, this is at the top of the cycle, and therefore it's called the citric acid cycle. So citric acid or tricarboxylic acid or the Krebs cycle, these all mean the same thing. We're talking about this cycle here. So citrate with 6 carbons, 1, 2, 3, 4, 5, 6, then is converted to isocitrate. Isocitrate gets decarboxylated, one of the carbons comes off, becomes a 5-carbon molecule, alpha ketoglutarate, 1, 2, 3, 4, 5 carbons, and NAD gets reduced to NADH in that reaction. Alpha ketoglutarate also gets decarboxylated, now becomes from 5 carbons to 4 carbons, CO2 is released. These are CO2s you breathe out, just like up here you had a CO2 you breathe out. And it becomes a 4-carbon molecule, succinyl-CoA, 1, 2, 3, 4, with CoA. Then that becomes succinate, and you get the one GTP being made if you're an animal cell, ATP being made if you're a plant cell. This is almost in like all intro bio books. It's called ATP because it's confusing if you introduce GTP and ATP. When you take cell phys, you're going to see GTP used a lot. You've already seen in this class how it's used in building up microtubules. It's a energy molecule. It's very similar to ATP. It just has a different base. Instead of adenine, has guanosine. That's the base. Succinate, 1, 2, 3, 4 carbons, gets rearranged to another 4-carbon molecule called fumarate, and then fumarate gets rearranged to another 4-carbon molecule, malate, rearranged again to another 4-carbon molecule, oxaloacetate, and then your second acetyl-CoA comes in and you repeat the cycle. So since you get 2 pyruvates per glucose, you would go around this cycle 2 times per glucose, once per pyruvate. Each of these enzymes are the enzymes that catalyze a particular reaction. So CAC1 is citrate synthase, then aconitase, isocitrate dehydrogenase, alpha ketoglutarate dehydrogenase, succinyl-CoA synthetase, succinyl dehydrogenase, fumarate hydratase, malate dehydrogenase. Citrate synthase and or isocitrate dehydrogenase are considered rate limiting. It depends on the book that you pick up. And most more modern books consider isocitrate dehydrogenase as the rate limiting step, but citrate synthase is a close second. You'll often see citrate synthase used as an indicator of aerobic activity. So if you are looking at a tissue and you want to look at how much aerobic activity is this tissue capable of, you can do a citrate synthase enzyme assay and how fast that enzyme can process oxaloacetate to citrate. is an indicator of how much of that enzyme is there, which is an indicator of how many mitochondria are active, an indicator of overall aerobic energy potential. Pyruvate, as we've said multiple times now, has to get from the end of glycolysis out here in the cytoplasm and then through the outer membrane and through the inner membrane to arrive at the matrix, where pyruvate dehydrogenase is going to convert it into acetyl-CoA. This, even though we knew that pyruvate dehydrogenase was in the matrix, didn't have a mechanism for how it got there until relatively recently, 2012. Okay, here's an H5P question for you. And here's an indication in your book that they recognize the pyruvate carrier discovered in 2012. As in glycolysis, as in any biochemical pathway, there are many points on that pathway where it can be allosterically regulated. Compounds that bind to the enzymes and regulate their activity. So pyruvate dehydrogenase is inhibited by acetyl-CoA, NADH, and ATP. So if you have a lot of those sorts of things, you wouldn't want to go through the cycle that makes those sorts of things, so you shut down that cycle. Whereas CoA, NAD+, and AMP, which are byproducts of breaking down acetyl-CoA, NADH, or ATP. If you have those breakdown products, that means that you don't have much ATP, NADH, or acetyl-CoA around, and so you would want to stimulate this pathway. So those are stimulatory, okay? And same sort of thing with different parts of the citric acid cycle.